Okay, so let's dive into something that might be like super relevant right now, especially if you're in Mr. Martin's IB biology class, and that's protein synthesis,
Yeah.
right? Now, when you first hear about protein synthesis, like it can feel kind of like overwhelming, I feel like.
Ew, God.
It's a lot of information, right? Like all these tiny pieces and how do they fit together?
It's like trying to learn a new language almost.
Exactly. Yeah, it's like all these new molecules.
All these molecular words.
Yeah, and they have like very specific interactions. And so in this deep dive, what we really want to do is sort of like break down those core ideas of protein synthesis and help you understand what are the most important things to remember.
Yes. And think about it as like a backstage pass. We're going to go behind the scenes of how your cells are actually building these essential proteins that do like pretty much everything in your body.
Yes, and specifically for Mr. Martin's class, we'll be kind of focusing on like the stuff that's covered in your IB biology materials.
Yeah.
So, think about transcription, translation, all that good stuff. All right, so where do we even begin with this process? Like,
Well, I think the best place to start is with like the big picture overview, right?
okay,
So protein synthesis is really a two-step process.
okay,
There's transcription and then there's translation.
got it.
So imagine it's like you have this really, really important master document
Okay.
and you need to make a copy to use somewhere else.
Okay, I like that analogy. So we're talking like DNA is our master document.
Yeah,
It's got all the genetic information, like a huge blueprint library.
it's the library.
And then let's say we want to build a very specific protein, right? So maybe a particular enzyme that does a very specific job in the cell.
Right.
We don't need the entire library.
No, you just need the one page.
We just need the instructions for that one enzyme. And that set of instructions is what we call a gene.
Exactly.
Okay, so how do we get that information out of the library and where it needs to be?
So that's where transcription comes in.
Okay.
Transcription is like going into the library and making a temporary portable copy of that blueprint. And the library in this case is the cell's nucleus.
Got it.
That's where the DNA is kept safe and sound. And this copy that we make is called messenger RNA or mRNA.
mRNA, okay.
And the amazing molecule that does this job is called RNA polymerase.
It's like our little molecular Xerox machine.
Exactly like a little photocopier in the library.
I love that.
So it makes a copy, and then once that mRNA copy is made, it can leave the nucleus and go out into the cytoplasm, which is like the main workspace of the cell.
Okay, so the mRNA has left the library with its blueprint. What happens next?
That's where translation comes in.
Okay.
So imagine now the mRNA is arriving at a construction site.
Okay,
This construction site is the cytoplasm of the cell.
got it.
And here, the actual protein building happens. The mRNA blueprint is read by these incredible molecular machines called ribosomes.
Okay.
And ribosomes are like the construction crew.
They've got all the tools they need.
They have all the tool, all the
Oh.
equipment, and they use the information in the mRNA to assemble the protein from individual building blocks called amino acids.
Okay.
Now, these amino acids are brought to the ribosome by another type of RNA called transfer RNA, or tRNA.
Okay, so this is like the delivery trucks bringing in the right materials to the construction site.
Exactly. It's bringing the right materials to the right place at the right time.
I love it. It's all coming together.
It was a well-oiled machine.
So to recap our two-step process,
Yes.
transcription is like making a copy of the DNA instructions for a specific protein
Right.
into that portable mRNA format in the nucleus.
Exactly.
And then translation is using that mRNA blueprint to actually assemble the protein using those amino acids and the help of ribosomes and tRNA out in the cytoplasm.
Perfect summary.
Okay, so let's dive a little deeper into transcription, that first step.
Okay.
Like what's actually going on in the nucleus when that mRNA copy is being made.
Right. So transcription takes place in the nucleus where the DNA is, and the key player here is that RNA polymerase.
Okay.
It's the one doing the copying.
So how does it know where to start copying?
Well, it doesn't just randomly attach itself to the DNA.
Right.
It binds to a very specific region called the promoter.
Okay.
And you can think of the promoter like a signpost on the DNA.
So it's like start here.
Exactly. It says attention gene starts here.
Okay, so RNA polymerase finds that signpost and then what?
Then it has to unwind and uncoil the DNA double helix.
Okay.
Because remember, DNA is this twisted ladder structure.
Right.
And to read the gene, it needs to separate those two sides of the ladder.
So it unzips it.
Exactly. Unzips it right at that specific spot.
And now we can see the bases.
And now the bases are exposed and ready to be copied.
Okay, so the DNA is open now. Which strand gets copied?
So only one of the separated DNA strands is used as the template.
Okay.
This is the master copy that the mRNA is going to be made from.
Okay.
And floating around inside the nucleus are the building blocks of RNA.
Okay.
These are RNA nucleotides.
And those are kind of like DNA nucleotides, but with one important difference.
One key difference, instead of thymine, RNA uses uracil.
Okay, so we got guanine, cytosine, adenine, and uracil.
Exactly.
So how do these RNA nucleotides know where to attach to the DNA template?
That's where complementary base pairing comes in.
Okay, I remember this from DNA replication,
It's the same principle.
like matching socks.
Yeah, like finding the matching pair.
If there's an adenine on the DNA template, a uracil RNA nucleotide will come in and pair up with it.
Exactly.
And guanine always pairs with cytosine.
And vice versa.
And RNA polymerase is like the facilitator, making sure those base pairs are lining up correctly.
Like the chaperone making sure everyone finds their dance partner.
Okay, and then RNA polymerase links those RNA nucleotides together to build that growing mRNA strand.
Exactly. It creates the chain.
Now, doesn't this building of the mRNA strand have a specific direction, like something about 5' to 3'?
Yeah, you're absolutely right.
I remember Mr. Martin talking about that.
For those of you in Mr. Martin's class learning about the higher level stuff, it's important to know that transcription happens in the 5' to 3' direction.
Okay.
So RNA polymerase adds the new RNA nucleotides to the 3' end of that growing mRNA molecule.
Okay.
It's kind of like reading a sentence from left to right.
Okay.
The molecular machinery also has a specific directionality,
Got it.
so it ensures accuracy.
So RNA polymerase is making this mRNA copy. What tells it, okay, stop copying, you've reached the end of the gene?
Well, the DNA sequence itself has what we call a terminator region.
Okay.
It's like the period at the end of a sentence.
It's the signal to stop.
Exactly when RNA polymerase hits that terminator sequence, it knows to stop transcribing.
Okay, so what happens to the mRNA and the original DNA once we've reached the end?
So the mRNA detaches from the DNA template.
Okay.
RNA polymerase lets go as well, and the DNA zips back up into that double helix structure.
Safe and sound back in the nucleus.
Back in the library.
And now our mRNA is a single-stranded molecule, carrying the genetic code ready to head out.
Ready for the next step.
Now, I know for eukaryotic cells, like our own cells, there's a little bit more that happens before the mRNA is ready to go.
Right.
Something about introns and exons.
Yeah, so in eukaryotic cells, the genes aren't like continuous stretches of coding information.
Okay.
They have these coding regions called exons.
Exons, okay.
And non-coding regions called introns.
Introns, got it.
So first, the whole thing, both exons and introns, get transcribed into this pre-mRNA molecule.
Okay.
It's kind of like when you film a movie and you have all the raw footage.
Okay.
Then you have to edit it.
Right.
You cut out the parts you don't need.
So the introns are like the bloopers?
Yeah, exactly. Like the extra stuff you don't want in the final movie.
Okay.
So in the cell, the introns are removed.
Okay.
And then the exons, which are the important parts, are spliced together.
Okay, so it's like editing a video to get the final version?
Exactly. And this final version is the mature mRNA.
And that's what's going to be used to make the protein.
That's the one that gets translated.
And there's also some extra stuff that gets added to the mRNA, right? Like a 5' cap and a poly-A tail.
Those are like protective caps and a reinforced end for the mRNA.
It's like giving it a little bit of armor.
Exactly. It helps it survive the journey out of the nucleus and into the cytoplasm.
Okay, so the mature mRNA with its instructions, it's ready to go.
It's ready to head out into the cytoplasm for translation.
Awesome. So let's talk about translation, that second step.
Okay.
So translation happens out in the cytoplasm, right?
Right in the main working area of the cell.
And specifically at ribosomes.
Exactly. Ribosomes are the protein factories.
So tell me more about ribosomes. What are they made of? How do they actually read that mRNA code?
So ribosomes are really incredible molecular machines.
I can imagine.
Each ribosome is made up of 2 subunits,
Okay.
a small one and a large one.
Okay.
And both of these subunits are made of ribosomal RNA, or rRNA, and proteins. And during the main stage of protein synthesis called elongation, the large subunit has these 3 really important binding sites for tRNA.
Okay, so tRNA, that's what's bringing the amino acids.
Exactly.
So what are these 3 binding sites?
Well, there's the A site, which stands for aminoacyl.
Okay.
That's where a new tRNA carrying an amino acid comes
What's
in.
the arrival gate?
Exactly. Then there's the P site, which stands for peptidome.
Okay.
That's where the growing protein chain is held.
Okay, so that's where the action's happening.
Yeah, that's where the chain is growing. And then there's the E site, which stands for exit.
Okay.
That's where the empty tRNA leaves the ribosome.
So it's like a little factory with an assembly line.
It's a very efficient system.
So the mRNA arrives at the ribosome. How does the ribosome know where to start reading the code to build the protein?
So the mRNA sequence is read in groups of 3 bases called codons.
Codons.
And each codon is like a specific inscription.
Okay.
It tells the ribosome which amino acid to add to the protein chain. Or it signals the start or the end of the process.
Okay.
So there's a special start codon, AUG,
AUG, all right.
which also codes for the amino acid methionine.
Okay.
And then there are 3 stop codons, UAA, UAG, and UGA.
So these are like the stop signs.
Exactly. They tell the ribosome to stop protein synthesis.
And this is where tRNA comes in, right? And delivering the right amino acid for each codon.
Exactly. tRNA is the key link between the mRNA code and the amino acid sequence.
Okay.
So each tRNA molecule has 2 important ends.
Okay.
One end is attached to a specific amino acid,
Okay.
and the other end has a sequence of 3 nucleotides called the anticodon.
The anticodon.
And the anticodon is complementary to a specific codon on the mRNA.
So it's like a lock and key.
Exactly.
Yeah.
The tRNA with the right anticodon will match up with the mRNA codon and deliver its amino acid.
Okay, so complementary base pairing is happening again this time between the mRNA codon and the tRNA anticodon.
We got it.
And the ribosome is moving along the mRNA, reading it codon by codon. And as each tRNA with the correct anticodon binds its amino acid is added to the growing polypeptide chain,
Yes.
and they're linked together by peptide bonds.
Peptide bonds hold the amino acids together.
So the chain keeps growing and growing.
It's the elongation phase of translation,
The protein is getting longer and longer.
according to the instructions in the mRNA.
And just like transcription translation also has directionality, right?
Yes. For the higher level IB students out there, translation also happens in the 5' to 3' direction.
So the ribosome is reading the mRNA in that direction.
From the start codon all the way to the stop codon.
Got it. Now this genetic code with its codons and amino acids, it has some interesting features, right?
Yes, it does.
Like why is it a triplet code? Why 3 bases for each amino acid?
Well, there are 4 different bases in RNA.
Right. Adenine, uracil, guanine, cytosine.
Exactly. And if each codon was only 1 base long,
Oh.
we could only code for 4 different amino acids.
Right.
But if codons were 2 bases long, we'd have 4 times 4,
Oh.
which is 16 possible combinations. Still not enough for the 20 amino acids that our bodies use to build proteins.
Right.
But with a triplet code, 3 bases per codon.
Yeah.
You have 4 times 4, which is 64 possible codons.
That's a lot more.
More than enough to code for all 20 amino acids.
Okay.
And it also provides some redundancy.
And that's what we call degeneracy, right?
Exactly. Degeneracy means that most amino acids are coded for by more than 1 codon.
Okay, so like multiple spellings for the same word.
Yeah, exactly.
Oh,
So there are different ways to spell the same sound.
So for example, the amino acid serine can be specified by different codons.
Right, like UCU,
Mhm.
UCC, UCA, and UCG all code for serine.
So if there's a mutation, a change in one base of the codon, it might not actually change the amino acid.
Exactly. That's one of the benefits of degeneracy.
That's pretty cool.
It's like a built-in safety mechanism.
And another interesting feature of the genetic code is that it's universal, right?
Yes, that's one of the most amazing things about it.
What does that mean exactly?
It means that the same codons code for the same amino acids in almost every living thing.
Really?
From bacteria to humans, we all use the same genetic language.
So a gene from a human could be read by a bacterium.
Exactly. And the bacterium could produce the human protein.
Wow, that's incredible. So it's like a universal language of life.
It really is.
And that's what makes things like genetic engineering possible.
Absolutely.
Like we can insert human genes into bacteria to produce things like insulin.
Exact. Bacteria can read our genes and make our proteins.
That's amazing. Okay, so back to translation. The ribosome keeps moving along the mRNA, adding amino acids to the chain until it hits a stop codon.
Right.
Then what happens?
So stop codons don't code for any amino acids.
Okay.
They're like the signal to terminate translation.
Okay.
So when the ribosome encounters a stop codon, special proteins called release factors come in and bind to the ribosome.
Okay.
And that triggers the release of the completed polypeptide chain.
So our protein is done?
The protein is released, and then the ribosome disassembles.
Okay.
It separates back into its small and large subunits.
And then the mRNA is released too.
Yes, the mRNA is free to go and maybe get translated again.
So we've got our chain of amino acids.
Yep.
Our polypeptide, is that a fully functional protein?
Not always.
Okay.
Sometimes the polypeptide needs further processing.
Like what kind of processing?
Well, it might need to fold into a specific 3D shape
Okay.
or have other molecules attached to it,
Right.
or even be cleaved into smaller active units.
Okay.
So it's like taking a raw piece of wood and shaping it into furniture.
Got it.
It needs those finishing touches to become functional.
Can you give me an example?
Sure, a good example is insulin.
Okay.
It starts as a larger inactive precursor molecule called preproinsulin.
Pre-proinsulin.
Yeah, and then it goes through several modifications.
Like folding and cleaving.
Exactly, to become the mature active insulin hormone.
Okay, so it's a multi-step process.
Many proteins require these extra steps.
Now, what happens to proteins once they've done their job or if they get damaged?
Well, cells have these amazing structures called proteasomes.
Proteasomes? Okay.
And proteasomes are like the cell's recycling centers for proteins.
Okay.
They break down proteins that are old, misfolded, or no longer needed.
So they're like the garbage disposal?
Yeah.
Kind of like that. And what happens to the amino acids?
The amino acids are released and recycled.
So they can be used to build new proteins.
Exactly. It's a very efficient system.
And that's important for maintaining a healthy proteome.
Absolutely. The proteome is the entire set of proteins in a cell.
So now let's talk about what happens if there are errors in the DNA mutations.
Okay.
How can mutations affect the proteins that are made?
So a mutation is basically a change in the DNA sequence.
Okay.
And one type of mutation is called a point mutation.
Point mutation.
And that's where a single base in the DNA is changed.
Just one base.
Yeah, just one.
And that can affect the protein.
It can, because remember, the DNA sequence determines the mRNA sequence.
Right.
And the mRNA sequence determines the amino acid sequence.
So if the DNA is changed, the mRNA might be changed.
Exactly. And if the mRNA is changed, the amino acid sequence might be changed.
Okay.
And if the amino acid sequence is changed, the protein might not fold correctly or function properly.
Can you give me an example?
Sure. A classic example is sickle cell anemia.
Sickle cell anemia.
This is a genetic disorder that's caused by a single point mutation.
Just one.
Just one base change in the gene that codes for hemoglobin.
Hemoglobin. That's the protein in red blood cells that carries oxygen.
Exactly.
Okay.
And in sickle cell anemia, this single base change causes a different amino acid to be put into the hemoglobin protein.
Okay.
So instead of glutamic acid, there's valine.
And that changes the shape of the red blood cell.
Yeah, it makes them sickle-shaped.
And that causes problems.
Yeah, it makes it harder for them to carry oxygen.
And it can cause blockages in blood vessels.
Exactly.
So this one tiny change in the DNA
Has a huge impact on the whole body.
Wow. It's amazing how much one little change can matter.
It really shows the importance of accuracy in these processes.
So just to recap our journey through protein synthesis.
Yes.
It all starts with the DNA in the nucleus.
Right.
Transcription makes a copy of the gene in the form of mRNA.
Mm-hmm.
The mRNA travels out to the cytoplasm and finds a ribosome.
The protein factory.
Translation happens at the ribosome with the help of tRNA delivering the right amino acids.
Based on the mRNA code.
And the ribosome builds a polypeptide chain.
Which may need further processing to become a functional protein.
And the sequence of bases in the DNA ultimately determines the sequence of amino acids in the protein.
Exactly.
And to bring it back to our analogy for Mr. Martin's class,
Yes.
remember Bob the Builder?
Yes.
DNA is his master instruction manual.
Right.
mRNA is that single page he photocopies for a specific project.
Mm-hmm.
And the ribosome is Bob himself using those raw materials, the amino acids brought by his helpers, the tRNAs, to construct that final rocket, the protein.
Love it.
Protein synthesis is like the foundation of life. It's how our bodies build everything.
Essential for all living things.
From enzymes to structural proteins, it's all thanks to protein synthesis.
Without it, we wouldn't exist.
And for Mr. Martin's class, think about this.
Yes.
We saw how a tiny change in the DNA, like a point mutation, can have huge consequences,
Right.
like in sickle cell anemia.
Mm-hmm.
So what other cellular processes rely on this same level of precision?
That's a great question to ponder.
What could happen if there are errors in those processes?
Lots of things to think about.
If you found this deep dive helpful, be sure to like this discussion.
Yes.
Leave a comment with any questions you have for Mr. Martin or for us.
We love to hear from you.
And subscribe to the Deep Dive for more explorations into the amazing world of biology.
Keep learning, keep exploring.
And until next time, stay curious.
Bye, everyone.